Welcome everybody in today's CellTalk presentation. Today, Dr. Vijay will talk about multiomic underpinnings of epigenetic aging and human longevity. Dr. Vijay is a senior scientist at Renix who leads bioinformatics, genomics, and next-generation sequencing research-related activities in the organization. He is currently working on the neoantigen cancer vaccine pipeline projects here. Vijay holds a PhD in Life Sciences from TMC Actrek, Homi Bhabha National Institute, Navi Mumbai. His PhD study focused on cancer genomics. Previously, Vijay was worked as assistant professor at graduate and postgraduate degree colleges as well. At Renix, he has gained experience in scientific, operational, and administrative matters. So let's welcome Vijay in for today's presentation.
Thank you, Dr. Nupur, for the introduction. Today, I have selected the paper that is Multiomic Underpinnings of Epigenetic Aging and Human Longevity, whose original author is Mauro Mattis et al. The title of today's presentation is A Multiomic Study Identifies High Confidence Genes Implicated in Biological Aging. The topic selected is longevity. The journal in which this paper was published is Nature Communications, which has an impact factor of 17, and this paper was published in 2023. This research has impact on aging and longevity. Introduction. Biological aging and aging in totality is a curiosity for scientists and people long. Everybody wants to live long and healthy life. Uh, and in the current situation, biological aging, it is an important healthcare issue, which increases morbidity, mortality, and healthcare cost in the population. So this is an important issue in the healthcare today. Aging-associated degenerative diseases, they cumulatively represent the leading cause of death in the developed nations and increasingly in the developing nations also. The molecular mechanisms of aging are poorly understood, and this is the reason why we are not able to effectively stop aging populations and manage the effects of the aging. So current medical interventions, because of this lack of understanding of aging mechanisms, is aimed at delaying the onset of and cure and treat the symptoms of individual age-related diseases. Those diseases or disorders that occur along with aging, those are only targeted for clinical intervention. There is an alternative way in which this can be treated is to pharmacologically targeting the biological processes that are responsible in the aging. For that, we need to identify those genes and pathways that are implicated in aging. And this is what this particular research paper is trying to achieve through a multi-omic analysis. General background. For some individuals, epigenetic age, it outpaces the chronological age. And what is referred to as epigenetic age acceleration. We all know there is a genetic and epigenetic aspects of our development and aging. So genes, they are stationary, they can be mutated, but overall which genes will be expressed at what moment in life, these are decided by the epigenetic factors. And therefore they have devised this term as epigenetic age acceleration, which is playing an important role in the aging process. So epigenetic age acceleration and human longevity phenotype, it comprises of multiple factors such as health span, lifespan, and exceptional longevity, and which is due to the multivariate factors. Therefore, it is been referred as multivariate longevity. Epigenetic age acceleration, it is associated with a number of health conditions and age-related diseases, which include substance use behaviors, alcoholisms or drug use, etc., atherosclerosis, and cancer and mortality. Epigenetic age acceleration, it can serve as an aging biomarker also. And large benefits could be achieved through interventions that slow the biological aging process, and especially if we can identify the epigenetic age acceleration related genes and pathways in the human body. So the present challenges for this particular field is to describe what are actually the biological correlates of epigenetic age acceleration. Can we identify and predict it? And then identify targetable biomolecules that modify the epigenetic age acceleration. Assess the biological similarities and differences between the epigenetic age acceleration and clinically relevant aging phenotypes like health span, lifespan, and exceptional longevity. The objective of the current research, it was to use multi-omic methods from different databases and integrate it with the integrated genomics, transcriptomics, and metabolic data to identify biological associations with four measures of epigenetic age acceleration in human longevity phenotype, which comprise health span, lifespan, and exceptional longevity. The methodology that was followed in this paper, it used multi-omic methods, which comparatively analyzed epigenetic age acceleration and a multivariate longevity-related phenotype, which comprised parental lifespan of an individual, health span of the individual, and exceptional longevity. Data from genome-wide association studies of four epigenetic clocks and a genome-wide association study meta-analysis of the components of multivariate longevity was performed. Then transcriptome-wide association studies was performed to identify transcriptomic associations with epigenetic age acceleration and multivariate longevity, which included novel epigenetic age acceleration associated genes. Then they employed fine mapping of the genes and then conditional analysis to prioritize genes which are high confidence genes with potentially causal relations with the aging related trends. Then the next step was the high confidence genes which were identified to the earlier studies. They were annotated with gene ontology categories. Then cis instrument Mendelian randomization, that is genes located in a particular specific location of the druggable genome, which is the genome in which the proteins which have are capable of binding to some pharmaceutical agent, and this is called as a druggable genome. So these databases, these are available. So these analysis were performed to identify potential drug targets that could identify the epigenetic age acceleration and multivariate longevity. The next step was to perform phenome-wide association studies to identify potential pleiotropic effects of promising genetic drug targets. The multiple effects that could be achieved with the genetic drug targets. The next study was metabolome-wide Mendelian randomization analysis for identification of circulating metabolites that impact aging-related traits. And the final step, cell type enrichment analysis was performed to identify which cell types were actually implicated majorly in the aging process. This slide depicts the methodology. So first, they identified the data sources to be used in the study, and these data sources were genome-wide association studies in which S&P phenotypes were associated with particular traits, and which include 34,710 data points of individuals, and also four epigenetic clocks, that is Grimm age, Pheno age, IEAA, that is intrinsic epigenetic age acceleration, and Hannum age. So these clocks were used. Then multivariate longevity database, which had 709,000 data points of individuals, and which comprised parental lifespan, health span, exceptional longevity, etc. Then they performed fusion transcriptome-wide association studies and expression QTL analysis. Then Nightingale Health 249 metabolic phenotypes data was used, which had 115,000 data points. And then at the final step, 120 cell types from tabula maris, there is a mouse database, it was used for carrying out this particular analysis. In the first step, for the objective identifying gene transcript and genetic drug targets for associated with epigenetic age acceleration was performed by using fusion pipeline and finding the drug targets by using Mendelian randomization. And after that, the significant genes were used to predict the gene function by using PRISM EXP database. Then for next objective, that is characterize the influence of circulating metabolites on epigenetic acceleration and multivariate longevity. They performed different types of Mendelian randomization studies like inverse variance weighted, Wald ratio, and Eger weighted, median weighted, and lasso Mendelian randomizations to find the potential associations. Then identifying the cell types highly expressed genes implicated in epigenetic age acceleration and multivariate longevity. They use select pipeline and magma database, SLDSC database to find which cells that are playing important role in the epigenetic age acceleration. This is the result of transcriptome-wide association studies, and these are the different Manhattan plots of different epigenetic clocks. That is IEAA, Grimm age, Hannum age, Pheno age. Those genes which are outside this threshold. These were considered to be significant associations, and these are the different genes which are showing statistically significant association with these particular phenotypes and uh, aging clocks. Here, the same analysis for multivariate longevity association was performed, and here we can see there are different genes which were found to be statistically significantly associated with multivariate longevity. And when a set analysis is performed for these four phenotypic locks and multivariate longevity, we can see that there are different genes which were found to be statistically significant, but there are some genes which are common across these different phenotype analysis. So here these three genes were found to be common with IEA and PhenoAge genetic clock associated genes. And here one gene and here one gene was found to be common. So these are the genes which were found to be high confidence. This means that if these are common across the different databases, then these are very high confidence genes. And this is the list of genes which enlist the different statistical metrics of why these genes are statistically significant. And there are also genes like TPMT here, IEA and PhenoAge, TPMT, and NHLRC1, which are occurring in multiple different associations. So these are again more high confidence genes which are implicated in epigenetic age acceleration. Now this result depicts the metabolome-wide Mendelian randomization analysis on multivariate longevity. Here, these in the blue they depict the genes which are metabolites which are negative, negative effect on the EAA, while in the green they have the positive effect on the EAA. That is epigenetic age acceleration. This is a result from select magma cellular association analysis, where they try to find out which cell types are actually implicated in the epigenetic age acceleration. So as you can see here, marrow precursor B cell, thymus, then marrow B cells, thymus immature T cells. So most of these cell types that they identified, these are immune related, actually immune cells or immune precursor cells, which were found to be statistically significantly associated with epigenetic age acceleration. And here the same genes were used for Mendelian randomization based association to identify immune cell traits with the aging phenotypes. And here they have used in these two phenotypes, multivariate longevity and IEA, and they use different Mendelian randomization methods to find the immune cells which are enriched in this association with the longevity or epigenetic age acceleration. So summarizing the results, 22 high confidence associations with epigenetic age acceleration and 7 with multivariate longevity were identified. Some novel genes were identified which were high confidence, such as plot1, KPNA4, TMX2, were found to be associated with epigenetic age acceleration. Then cis instrument Mendelian randomization of the druggable genome that associated TPMT gene and NHLRC1 gene with the epigenetic aging. Metabolomics Mendelian randomization analysis identified a negative effect of non-high density lipoprotein cholesterol and associated lipoproteins on the multivariate longevity. Cell type enrichment analysis implicated immune cells and precursors in epigenetic age acceleration and multivariate longevity. Discussion. Novel genes were identified with the associated with the epigenetic age acceleration. Then transcriptomic effects of previously identified gene associations with epigenetic age acceleration and multivariate longevity were elucidated. Then several identified genes were previously implicated in previously implicated in age-related endpoints were also identified, and which were also implicated in cancer. Shared genetic control of aging and the immune system was identified, and which highlights the need for further research into the role of immunity and inflammation in the development of age-related diseases. Which is a very important association that they have found. Transcriptome-wide association studies identified many genes that have previously been linked with aging and age-related diseases, which supports the validity of the approach used in this particular study. This study also has some limitations. So the genome association studies and UK biobank participants' parental lifespans, they may reflect the common cause of health and disease in the UK from several decades ago, because there is a huge time gap between the parental generation and the generation that is being studied right now. So those conditions are shifted now. So those conditions are not similar in the parental generation and the people which were part of this particular study. The human data sets used throughout the study almost exclusively comprised of the participants of the European ancestry. Due to differences in the allele frequency, LD structure, and the genetic architecture of the complex trait between the ancestral populations, multi-omic analysis have limited transnational generalizability. Cross-tissue gene expression weight dataset did not include expression weights for all genes that may be relevant to the epigenetic aging or longevity. Due to the limited availability of broad human single-cell RNA sequencing datasets, musculus single-cell RNA-seq data was used for cell type enrichment analysis. So all these immune cells that were identified, this should be confirmed by using human single-cell RNA-seq data. Concluding, the study identified transcriptomic, metabolomic, and cellular correlates of epigenetic age acceleration. This study also uncovered clinically relevant multivariate longevity phenotype, also prioritized biological pathways relevant to aging. This study also identified drug targets that may reduce epigenetic age acceleration and promote longevity. It also identified similarities and differences between epigenetic aging and multivariate longevity, and between different epigenetic clocks were identified. Identified similarities and differences between different epigenetic clocks also. These findings may provide valuable insights into the transcriptomic, metabolomic, cellular immune-related architecture of age-related phenotypes, and may guide future research aimed at characterizing and pharmacologically combating pathological aging. Impact of the research: the results from this study sheds light on druggable targets and biological pathways, which involves aging, epigenetic clocks, and human longevity. These results will be helpful to support the future research efforts aimed at delaying or potentially stopping human aging. Thank you for your kind attention.
Thanks, Vijay. So we have time for some questions. You can type your questions in the chat box. I don't find any question in the chat box. So I think we can thank Dr. Vijay for this nice talk. Thank you, Vijay.
Thank you, Dr. Mukul.
Our next presentation will be given by Jai Darji. He will present on systematic evaluation of machine learning algorithms for neuroanatomically based age prediction in youth. Let's join for the presentation of Jai on 3rd of April 2024. Thank you everybody for attending today's CellTalk.